In biology, cell culture is the process by which cells are grown in controlled conditions outside of their native environment. This technique has enabled scientists to use cells, the fundamental unit of life, as an imperative model system for understanding physiological processes in the human body. History of cell culturing. One of the major contributions of cell culturing is the development of polio vaccines. Back in the early 1930s, researchers had to use live animals to grow poliovirus. But with cell cultures, researchers were able to control virus production, which eventually led to the creation of the vaccine. Fast forward to the 1970s, mammalian cell culture was used to create properly folded and glycosylated proteins, such as interferons and antibodies. They were impossible to make in prokaryotic E. coli cells because E. coli cells lack post-translational modifications. Nowadays, cell culture is being used widely in laboratory settings, particularly in the fields of vaccine research, cancer research, and protein therapeutics. Categories of cell culture. Cells that are used in laboratories can be generalized into two categories: primary cells and established cell lines. Primary cells are cells that are directly prepared from an organism's tissues. If cultured under the right conditions, primary cells may grow and proliferate, but they are only able to do so a finite number of times. This is because each time a cell divides, part of its DNA telomere is lost. And after a certain number of cell divisions, the primary cells would reach senescence, a permanent stage where the cells lose the ability to divide and grow. Established cell lines, on the other hand, are able to escape normal cell cycle constraints and grow indefinitely, making them extremely useful for long-term research. Whereas primary cells are obtained directly from donor tissues, cell lines can be derived from clinical tumors or created from transforming primary cells with viral oncogenes or chemical treatments. These reagents or treatments target one of the checkpoints in the 5 phases in the mammalian cell cycle. Cell cycle checkpoints are surveillance mechanisms in eukaryotic cells that ensure the proper progression of the cell cycle. There are 3 main checkpoints in the cell cycle. The first checkpoint is located at the end of G1 phase to ensure the cell is ready for DNA synthesis. The second checkpoint is located at the transition from G2 phase to M phase and verifies that DNA replication in the S phase has been completed successfully and that there is no DNA damage. Finally, during mitosis, there are several checkpoints to ensure that the cell is in a proper position to complete cell division. When cells start growing uncontrollably, it is because they have bypassed one or many of these checkpoints. Many viral oncogenes, also known as tumor suppressor genes, such as SV40, inactivate proteins associated with the cell cycle checkpoints, thereby disrupting the cell cycle regulation. Experimental considerations. While both the primary and immortalized cell types are widely used in research settings, how do you choose which one to use? The decision depends on your research goals, and in some cases, you might need both. For example, in drug development and testing, primary cells such as human hepatocytes are the preferred choice for toxicity testing of new drugs, as they provide physiological relevance. However, while primary hepatocytes replicate liver metabolism in vivo, the limited lifespan and expensive procurement processes can make large-scale studies extremely challenging in vitro. This is why immortalized cells are often used in high-throughput screening of drug candidates. Immortalized cells' ability to proliferate indefinitely and grow in large quantities can help narrow down drug candidates, even though they may not fully replicate the biology of primary cells. In addition to physiological relevance and growth characteristics, other factors such as genetic and phenotypic stability, ethical consideration and cost also play a role. For genetic research, immortalized cell lines are commonly used as they provide a consistent genetic and phenotypic profile. In addition, cell lines also provide an opportunity for scientists to study the downstream effects of gene overexpression or silencing. From a cost perspective, while a small amount of primary cells is cheaper than immortalized cell lines, it can still put a constraint on your budget if large amounts or repeated experiments are required. Considering the longevity of immortalized cells, it may be the more cost-effective option for lengthier experiments. Future directions. The choice between primary and immortalized cells should be guided by the specific goals of your research. Primary cells offer more accurate models of in vivo biology, but come with challenges in terms of growth, variability, and cost. Immortalized cells provide convenience, consistency, and scalability, but may not fully capture the complexity of natural cellular behavior. Today, as a pioneer of cell immortalization in the industry, ABM uses several methods to make our immortalized cells more closely resemble primary cells. This includes using conditional immortalization methods, as well as specific immortalizing agents like hTERT instead of viral oncogenes. With this, immortalized cells have the potential for broader use in translational research and certainly in practical medicine in the future. To learn more about the basics to cell immortalization, visit our knowledge base at the link below. Also be sure to check out our next video. Please leave your questions and comments below, and we will answer them as soon as possible. Thank you for watching.